Hello, everyone. This is me again, Camila Pimentel, and this is an introduction to chromosomal abnormalities. You may need to watch my videos on mitosis and meiosis to understand this one better. So, let's begin. A chromosome is a structure inside the nucleus of a cell that contains genetic information made of a tightly coiled molecule of DNA wrapped around proteins which replicate in both mitosis and meiosis. To learn more about these processes, watch our videos on them. Chromosomes have a short arm called P, a long arm called Q. Telomeres at the end, which get shorter the more the cell replicates, become a marker for aging, and a centromere, which is the narrowest part of a chromosome and also where they attach to sister chromatids. Humans have 22 pairs of homologous autosomal chromosomes and one sexual pair, which can be XY or XX. XY for genetic males and XX for genetic females. Although sex is a little more complicated than that, that's out of the scope of this video. Chromosomes in the karyotype are ordered by size and they can have numerical and structural abnormalities. This is an example of a disorder in the segregation of chromosomes during meiosis. This can cause both a monosomy and a triploidy depending on if the cell number 1 becomes a zygote or cell number 2 becomes a zygote, respectively. This can happen in autosomal and sexual chromosomes during mitosis or meiosis. Errors in chromosome replication, exposure to certain environmental factors, and parental age are risk factors. These alterations have significant clinical consequences. They account for about half of spontaneous abortions and can also cause stillbirths, neonatal deaths or hospitalizations, malformations, intellectual disability, or an identifiable syndrome. If abnormalities occur in somatic cells during mitosis, then you can get something called mosaicism, where only one group of cells is affected, with variable effects. In mosaicism, one group of cells can be normal, while the other can be abnormal. It has variable effects because it's pretty much an individual to individual situation, as no mosaicism is the same. Aneuploidies is any number of chromosomes that's not 23 pairs in humans. This happens when homologous chromosomes fail to separate and they end up in one daughter cell together, while the other cell will have one chromosome missing. Disorders vary depending on if the cell is in a sexual cell or a somatic cell. If it's in a somatic cell, it originates mosaicism, but a sexual cell in meiosis can result in triploidy, three copies of what's supposed to be one pair of chromosomes, monosomy, a single copy, or in rare cases, tetraploidy, with four copies instead of the usual pair. Let's use Down syndrome as an example. It's classically represented as a numerical abnormality in the pair 21. They have three instead of the pair. Structural abnormalities can result from breakage and incorrect rejoining of chromosomal segments. A range of structural chromosomal abnormalities result in disease. Structural rearrangements are defined as balanced if the complete chromosomal set is still present, though rearranged, and unbalanced if information is additional or missing. Translocations are especially common amongst the chromosomes 13, 14, 15, 21, and 22 because they cluster together during meiosis. Unbalanced translocations are deletions. A chromosomal deletion is the loss of a segment of a chromosome, which removes genetic material and can vary in size. Duplication. A chromosomal duplication is a type of mutation where a part of a chromosome is copied, resulting in extra genetic material in the form of an extra copy of a chromosomal segment. Insertions. An insertion is the addition of one or more nucleotides into a DNA sequence or a larger segment of one chromosome being inserted into another. Ring chromosomes can result when a chromosome undergoes two breaks and the broken ends fuse into a circular chromosome. An isochromosome can form when an arm of the chromosome is missing and the remaining arm duplicates. It happens when the centromere divides horizontally versus vertically. Robertsonian translocation. In this case, two acrocentric chromosomes that have centromeres close to the edge of the chromosome break at the centromere, losing both of their short P arms and creating a new single chromosome containing one centromere and the Q arms of both original chromosomes. All of the above can cause monosomies or triploidies, even if they don't have one chromosome fully gone or duplicated. This is because the genetic information gets duplicated or goes missing. Although the degree can vary depending on how much is gone or how much is added. A balanced translocation is a chromosomal rearrangement where segments of non-homologous, not to confuse with sister chromatids, chromosomes are exchanged without a net gain or loss of genetic material, making the carrier generally healthy but potentially passing on unbalanced chromosomes to offspring. A disease can arise as a result of a balanced rearrangement if the breaks in the chromosomes occur in a gene, resulting in an absent or non-functional protein, or if the fusion of chromosome segments result in a hybrid of two genes, producing a new protein product whose function is damaging to the cell. Testing. Prenatal screening and testing can be performed to examine the chromosomes of the fetus and detect some, but not all, types of chromosomal abnormalities. Why is testing important? In countries where abortion is legal, uh, parents may choose to go that route. It's also good to know what to expect so parents can prepare in advance, both psychologically and practically, to receive a child that could have significant health issues like mental and physical disabilities, or that are at increased risk of cancer, amongst other diseases. In amniotic fluid samples, karyotyping is considered the gold standard for diagnosing fetal aneuploidies. It examines the size, the shape, and number of chromosomes in a cell. Fish. This method uses fluorescent labeled DNA probes to assess the present location and copy number of the complementary DNA sequence of interest in an individual genome by hybridization. This technique is particularly advantageous as a relatively rapid diagnostic tool for structural chromosomal abnormalities involving small segments of DNA, which can be microdeletions, translocations, etc., and for detecting trisomies due to a more distinct fluorescent reading. I want to end this by saying that every life is important, but if you don't feel capable of bringing a child to this world with significant health issues, I don't blame you. Your body, your choice. It could be argued that a special needs baby could suffer in an unprepared household, and the adoption system is biased towards able-bodied individuals. Not only that, children with special needs grow up to become adults with special needs and may outlive your care. Now, it's not to say I prefer abortion over giving them a chance at life. I just want to say that I don't blame parents that take that route. If you are a parent or have someone close by with a chromosomal disorder with important health implications, I admire your endurance and let me tell you that you're not alone. There's non-lucrative foundations everywhere over the world, and you may find one that fits your person's needs so you don't get burnout. Make sure to take care of your own mental health and physical health before you take care of others. Your, your special person is important, but so are you. You are important. I want to give a shout out to Langman's Medical Embryology, which is the book I'm using to base the order of my videos. Also, the two articles, Chromosomal Disorder by Encyclopedia Britannica and the appendix Chromosomal Abnormalities by Genetic Alliance of their book Understanding Genetic, a New York Mid-Atlantic Guide for Patients and Health Professionals. These are the credits. These are the credits: Camila Pimentel, drawings, editing, script, voiceover; Jezebel, script, clarity; Joanna Strange, humanitarian approach; my family and dogs, moral support; and finally, you that's watching this video. I want to thank you for making it to this point. I have received immense support, more than I thought, with less than a month of creating this channel, so I can't stress enough how grateful I am. If it's not too bothersome, please subscribe, give a like, and leave a comment below. Thank you.